So hi, today is the eighth problem solving session for the course Experimental Biotechnology. The course instructor is Professor Vishal Trivedi from IIT Guwahati and I am Shrija Ghosh, a PMRF from IIT Kanpur. Uh, so today we'll discuss a new topic which is about cell culture medium and cell fractionation. So we'll see different types of media that are used for different types of cell culturing. What are the different types of cells present that we use in biotechnology, and we'll use different methods how to isolate uh, different parts of the cell through various techniques. Okay, so we'll be discussing few questions regarding these topics, and if you have any kind of doubt, you can stop me at any point and ask me, or you can write it in the chat box. These questions will help you solve your uh, assignments that are given for this week. Okay, so I'll move to the first question. Question number 1. What is a major structural difference between prokaryotic and eukaryotic cells in terms of their DNA? Option A, prokaryotic cells have linear DNA while eukaryotic cells have circular DNA. Prokaryotic cells have multiple chromosomes while eukaryotic cells have single chromosome. Option C, prokaryotic cells lack DNA while eukaryotic cells have DNA in the form of plasmids. Option D, prokaryotic cells have a single circular DNA molecule while eukaryotic cells have multiple linear chromosomes. So, what is the major difference between the prokaryotic and eukaryotic cells? Yes, so yes, you have answered fourth option. That is the correct option. So structurally, the major difference between the DNA of a prokaryotic cell and a eukaryotic cell is that prokaryotes have a single circular DNA molecule, whereas eukaryotes have the linear DNA, which is organized in specialized structures called chromosomes. So fourth option is the correct answer. So for prokaryotic cells, here I have listed the difference between the prokaryotic and eukaryotic DNA. The DNA is circular and double-stranded for prokaryotic cells, and for most eukaryotic cells, the DNA is linear and double-stranded. In prokaryotic cells, we don't have any compartments. Okay, so the cell is just one large room, and in eukaryotic cells you can say it is divided into multiple compartments. It has multiple organelles, so different membrane bound organelles are present. So if I just have to draw a rough diagram for prokaryotic cell, so a prokaryotic cell will have just one room. This is the membrane, so we can have two layers of membrane, outer membrane and inner membrane, and then everything is present. So this will be the cytoplasm. The inner part of the cell will be the cytoplasm, and everything, the DNA will be present in the center in a localized region called nucleoid, but there's no specialized structure called nucleus. In eukaryotic cells, we will have multiple membrane bound organelles. So we will have a nucleus which has its own membrane. So this is the cell membrane, the outer cell membrane. Okay, and then inside the eukaryotic cell you will have different organelles with its own membrane. So this is nucleus. This will be the membrane of the nucleus, the nucleus membrane. Okay, inside the nucleus we will have the DNA. Okay, for eukaryotic cell. So DNA is enclosed in a membrane bound organelle, whereas in prokaryotic cells the DNA is just floating in the cytoplasm. Okay, so it floats freely, whereas in eukaryotic cell it is present inside the membrane and it is associated with various proteins. These such proteins are called histones. Okay, so these proteins basically uh, ensure that the DNA is well packaged. And when you will study about different processes, how the genes are uh, translated in the cells, how the different proteins are produced inside the eukaryotic cell, you will understand why these proteins that are present with the associated with the DNA is important. Okay, and then we have the DNA packaged into chromosomes. So basically, these chromosomes are further associated with these histone proteins. And in prokaryotic cells, the DNA is less organized. The DNA is just floating in the cytoplasm freely. It might be associated with one or two protein, but those proteins are not uh, making it in like folding it in any organized structure or anything. Okay, so it is just floating freely in the cytoplasm. Since prokaryotes don't have any organelle, so anything that is present inside the prokaryote is present in the cytoplasm only. Whatever proteins are there, they are present in the cytoplasm, or they are embedded in these cell membrane. But in but in eukaryotic cells, everything is present in particular organelle. So we can have mitochondria, and we can have a nucleus, we can have other organelles, different vesicles, lysosomes, and everything. Okay, so sorry, liposomes and everything. So different kinds of organelles are present in eukaryotic cell, but in prokaryotic cell only a single compartment is there. So you can think of a single room for prokaryotes, and for eukaryotes you can think of it like an apartment where you have multiple rooms, like a 3 bhk 4 bhk apartment. Okay, so moving on to the next question now. Question number 2. Carbohydrates in the growth medium is used for dash in the cells. Option A, energy generation. Option B, nucleotide synthesis. Option C, amino acid synthesis. And option D, protein synthesis. Carbohydrates in the growth medium are is used for dash in the cells. Yeah, you have answered option 1, that is energy generation. Yes, that is the correct answer. So carbohydrates, mainly the most common carbohydrate is sugar. Okay, so here I have different components of a growth medium. So for any cell to grow, you can imagine, like, human beings are also composed of cells, right? Now, if we need to grow, what do we need? We need to we need components that can be used for synthesis of the genetic material, that is DNA or RNA. Okay, so we need things that can make nucleotides. Then we need fatty acids. Why do we need fatty acids? Because all the membranes are composed of fatty acids, so lipids and fatty acids are important. Then we need proteins, because all the proteins that is present in the body, it has to come from somewhere. So source of protein or source of amino acid, like amino acid synthesis is necessary. Amino acids are the building blocks of protein. So once amino acid will be there, they can join together to form a protein, right? And then comes carbohydrate. Carbohydrates, basically, major source of carbohydrate is sugar. So any form of sugar gives us energy. Okay, so carbohydrates like sugars, they give us energy. So here you can see this shows the elements, different elements that are gained from these different sources that we provide in the growth medium. So if we are adding any amino acid, basically they are acting as the building blocks for proteins. Okay, and the elements that are present are carbon, hydrogen, oxygen, nitrogen, and sulfur. Then when we are supplying any sugar, sugar is majorly composed of carbon, hydrogen, and oxygen, and it will be used up for energy. Then when we give any lipid or fatty acids in the growth medium, we are basically giving carbon, hydrogen, oxygen, phosphorus, and sulfur. And for nucleotides, we are giving carbon, hydrogen, oxygen, nitrogen, and phosphorus. So if you see, if I make a growth medium where I am giving carbon, hydrogen, oxygen, nitrogen, sulfur and phosphorus, you can imagine that that growth medium will be like should be able to synthesize all these components, and as we are providing all the elements. But of course we need other growth factors like different vitamins and uh, other growth factors, since those factors are basically associated with the enzymes. So let's say amino acid synthesis. Okay, so if I give the cell carbon, hydrogen, oxygen, nitrogen and sulfur, they can make the amino acid, but then amino acid biosynthesis pathway will have certain enzymes which have uh, cofactors that are derived from different vitamins which cannot be synthesized by the cell. So for that purpose we will have to provide these growth factors or cofactors uh, along with these elements. Okay, so and for carbohydrates, if you know that if you know the glycolysis pathway, so whenever we provide any sugar, let's say the simplest sugar, that is glucose, through glycolysis the glucose is converted into pyruvate, and then later this enters the Krebs cycle in the form of acetyl coenzyme A that can uh, generate enough NADPH or NADH, which can enter the electron transport chain to produce ATP, right? So from glucose, so ATP is the energy source. If you remember from your school days, you must have studied that the mitochondria is the powerhouse of the cell, right? So in eukaryotic cells, mitochondria produces these ATP. So ATP is the energy molecule. So basically why it is called a powerhouse of the cell? Because it is producing the power by generating more and more ATP. So carbohydrate or whatever sugar you are uh, taking in. So if you are having complex sugar like sucrose or fructose, so those sugars will ultimately either incorporated into the glycolysis pathway or they will be broken down. If it is a complex sugar like sucrose, it will be broken down into glucose units and then they will be incorporated into the glycolysis pathway. So this pathway is called glycolysis. Then this pathway is the TCA cycle. TCA cycle or the Krebs cycle. It is also called Krebs cycle. Okay. Then this is the electron transport chain, so ETC. Okay. So ultimately any carbohydrate that is taken up is converted into ATP, which is the source of energy. That's why we say carbohydrates are used for generating energy. Okay. Okay. Let's move to the next question now. Which of the following is an example of a defined medium or a synthetic medium? Option A, nutrient agar. Option B, tryptic soy broth. Option C, LB broth. And option D, minimal salts media. Which of the following is an example of a defined or synthetic medium? You have answered option 4, that is minimal salts medium. That is the correct answer. Option D, minimal salts medium. So we have two terms, defined medium and uh, complex medium. So synthetic medium or defined medium, by the name itself you can see it is defined, right? So the composition of this medium is known. What do you mean by composition is known? What do we mean by composition of the medium is known? That means the amount, all the elements that we are adding, the amount of carbon, nitrogen, hydrogen, oxygen, the elements that I showed in the previous slide. So amount of all the elements, essential elements, whatever metal ions you are adding, uh, or if you are adding any vitamins. So for everything, the amount of each element is known. Okay, so amount of element is known. So that makes the medium defined. Okay, so that basically in the minimal salts medium, what we are adding only different kinds of salt. So we are adding, let's say, ammonium chloride for nitrogen source. Then we are adding some carbon source like glucose, let's say, or sucrose. Then so you know the formula of glucose, so you know how much exact. So if I am adding certain moles of glucose, I know how many moles of carbon has been added to the medium. Okay, so if I'm adding sodium chloride, I know how many moles of sodium and how many moles of chloride is present. Okay, now for metal ion, if I'm adding copper chloride or iron chloride or iron sulfate, so I know how much amount of iron, how much amount of sulfur, and how much amount of oxygen is added to the medium. So that is called a defined medium. Now, in case of nutrient agar, tryptic soy broth, or LB broth, we add some components like uh, peptone or tryptone. Then we add things like yeast extract. Okay, so these things are basically derived from different animal or eukaryotic source. So yeast extract, by the name itself, you can understand that it is derived from yeast. So basically it's a in very simple language, you can understand that it's a paste of yeast. Okay, think of it like that. So since yeast has multiple growth factors and uh, components present inside it, so we can actually inactivate that yeast and get an extract out of it and use it in the growth medium. So all the factors will be present, but the exact amount of each cofactor in the yeast extract will be different. Okay, so every batch of yeast extract, if I open two bottles of yeast extract, they both might have different elemental composition. Okay, so that means when we are adding these yeast extract or tryptone, or in the tryptic soy broth, we generally add extracts of soya bean and casein powder. So these are different kinds of protein. Soya bean is a protein source, right? So if we are adding extract of soya bean, that means we are adding a protein source, a nitrogen source, or a carbon source. But this source, we cannot tell how many nitrogen, like what is number of moles of nitrogen present in the soya bean extract. Okay, so one soya bean can have different composition than the other. So if you are taking some 100 grams of soya bean and taking the extract out of it, you cannot really tell the exact elemental composition. So all these types of media, nutrient media, LB broth, tryptic soy broth, these are basically complex medium, because we don't know the exact elemental composition of these media. Whereas minimal salts media, we know the exact composition for that media. Okay, I hope that's clear. So although you are answering it correctly, I'm explaining it for the sake of other people who will be watching this video to understand. Okay, so, and if you also have any doubt in the explanation part, do let me know. Okay, I'll move to the next question. Question number 4. Dash is added to DMEM mammalian cell culture media as a buffering agent. Option A, sodium bicarbonate. Option B, ammonium sulfate. Option C, sodium phosphate. And option D, potassium phosphate. What is added to the DMEM mammalian cell culture media as a buffering agent? Option A, sodium bicarbonate. Option B, ammonium sulfate. Option C, sodium phosphate. And option D, potassium phosphate. You have answered option A, sodium bicarbonate. Yes, that is the correct answer here. So sodium bicarbonate. So in the DMEM is the most common uh, medium that we use for mammalian cell culture, and we add sodium bicarbonate in the media as a buffering agent. What do we mean by buffering agent? Buffering agent is an agent that can actually help maintaining the pH of the solution. Okay, so to maintain the pH of the solution, pH of the medium. Now, why do we have to maintain the pH of the medium? So for any cell to grow, it will be growing in a particular pH. It will be growing at a particular pH. So mammalian cells basically cells derived from any mammal. So it can be human cell line, it can be some hamster cell line or mouse cell line. Okay, so any cells that are derived from a mammal. Now, if you know, for humans, our cells can only grow at a certain pH, and in our body has a mechanism to maintain that pH. Okay, and our cells will grow in that particular pH only. Now, when we are culturing these cells outside, what do we mean by culturing these cells outside? We are basically uh, someone has made a cell line out of these cells. So let's say skin cells or ovary cells. Okay, so all the most of the cells that are present in the human body or that are present in some other mammal, uh, they have been converted into a cell line where they can grow for certain number of generations. Like they can grow easily. Okay, so when the cell line is made, now we have to provide growth medium, because I told you that you need energy source, you need protein building blocks and different things. So you have to provide all those elements that I showed. So when you are providing such elements and when the cell is growing, there's a chance that cell is releasing kind of some kind of component that can reduce the pH. Or when the cell is growing, there will be some cells that are dying along with the cells that are dividing. There will be cells that are dying. Once the cell dies and get lysed in this medium, the cell components come out in the medium, and those components can make the uh, medium acidic. Okay, so to prevent the pH from decreasing, to prevent the pH from going towards the acidic range, to maintain the pH of the medium, we add sodium bicarbonate. And what does sodium bicarbonate do? So when the cells are growing, the cells are obviously respirate, uh, like the cells are also living in the medium, right? And we always culture mammalian cells in a CO2 incubator. Okay, CO2 incubator, that means we are providing 5 to 10% of 5 to 8 generally uh, percentages provided. So we are providing CO2 to these cells. So what does the CO2 do? The CO2 dissolves in water that is present in the medium, and becomes carbonic acid. Okay, and this carbonic acid and sodium bicarbonate, so you know how a buffer is made, right? Buffer is made with a weak acid and its conjugate salt. Okay, so if you mix a weak acid and a conjugate salt, you will get a buffer, right? So here carbonic acid is the weak acid, right? So carbonic acid is the weak acid, and we are adding sodium bicarbonate, that is the conjugate salt, and they both will together they both will act as a buffering agent, and they will help maintaining the pH. That means any small amount of acid or base when added to the medium, the pH will not change. Okay, so any small amount of acid or acidic components that are released by the cells or that are released during the death of the cell will not affect the pH of the whole medium, because we have a buffer present in the medium. Okay, so that is the purpose we add sodium bicarbonate in the mammalian cell culture medium, because it can mix with the carbonic acid and create a buffer system. Okay, so now I'll move to the next question. Question number 5. Dash is added to the DMEM mammalian cell culture medium as a growth factor. So now we saw as a buffering agent. Now this question asks for a growth factor. What is added to the DMEM mammalian cell culture medium as a growth factor? Option A, DMEM. Option B, fetal bovine serum. Option C, sodium bicarbonate. And option D, yeast extract. So what is added to the DMEM medium as a growth factor? So what do we add in the DMEM medium as a growth factor? Option A, DMEM. Option B, FBS, that is fetal bovine serum. Option C, sodium bicarbonate. And option D, yeast extract. Yeah, option B, you are answering fetal bovine serum. Okay. We have 2 more people who have joined the class. Do you want to answer this question? So option B, fetal bovine serum is the correct answer. So as I explained, for buffering agent, we are adding sodium bicarbonate. Similarly, for providing different growth factors. So what are these growth factors? All kinds of vitamins and different cofactors that are provided from the fetal bovine serum. So what is serum? Serum is basically the liquid part of the blood. So serum is basically what you get when the blood gets clotted. So if when you have blood, and if you keep the blood in a vial, so once everything is settling down, the top part that you will get is serum. Okay, now this serum has certain cofactors and certain components that can be used by the cell for its growth. So I told you about the different components that are required for required in a growth medium, all the elements, carbon, hydrogen, oxygen, nitrogen, right? Along with that, I told you we have to provide extra growth factors. Those growth factors in a mammalian cell culture is provided by the fetal bovine serum. Okay, so option B is the correct answer. Moving to the next question. Mammalian cell culture medium is filtered and not autoclaved because option A, the glucose present in the media might get charred. Option B, the growth factors are heat sensitive and might get degraded. Option C, filtration is cheaper. And option D, media components react with each other inside the media. So why do we not autoclave the mammalian cell culture media, and why do we filter it? Option A, glucose present in the media might get charred. Option B, the growth factors are heat sensitive and might get degraded. Option C, filtration is cheaper. And option D, media components react with each other inside the media. So, why do you think it is generally filtered? Yeah, Shaina has answered option B. The growth factors are heat sensitive and might get degraded. Okay. Anyone else wants to answer? Come on, take a guess. If you don't know the answer, I'm anyway going to explain the answer. Why do you think mammalian cell culture medium is not autoclaved and it is filtered? So option B, what China has answered, is the correct answer. Growth factors are heat sensitive and might get degraded. So what is the purpose of filtration or autoclaving? First, let's discuss that. So we do these processes to sterilize the medium. So these are process for sterilization. Okay, these are process for sterilization of the medium. What do we mean by sterilization? Removal of any living cell or the spores of the living cell. So removal of anything that can grow in the medium on its own. So basically, we are making this medium to grow a particular cell that we will inoculate. So we don't want any contamination from outside and any contamination in the medium. Like if any cell is already present in the medium, because we are making the media in water, and water can have certain things, and we are making the media in open air. So a lot of microorganisms are already present in the open air. So we need to remove those microorganisms that are present in the surroundings in order to uh, culture a particular cell inside the medium. So for that, the removal of any living particle is called sterilization. So these are two common methods, filtration and autoclaving. So in autoclave, what we do, we heat the medium at 121 degrees Celsius under pressure. So we apply 15 psi pressure. So when we apply 15 psi pressure, uh, the temperature that is achieved is 121 degrees Celsius. So this is steam sterilization, and we maintain this temperature for 15 to 20 minutes, considering that in this particular time all the living particles that are present in the media gets degraded and they get killed, and that's how we get sterile medium. In filtration, what we do, we generally pass the medium through 0.22 micron filter. Okay, why 0.22 micron? Because any living particle that is present in the environment is generally bigger than 0.22 micron. The size of the smallest bacteria is more than 0.22 micron. So when we pass the media through 0.22 micron, no microorganism can pass through the filter and they are trapped on the filter, and that's how the filtrate that we get is sterile. Now, in mammalian cell culture, we discussed fetal bovine serum, right? In the previous question, we discussed fetal bovine serum. So, fetal bovine serum has certain growth factors. Those growth factors are actually heat sensitive. So, I told you in autoclave, we are heating the media up to 121 degrees Celsius. So, it's a very, very high temperature, right? It's beyond boiling point of water. And we reach this temperature by under pressure. When we heat the water under pressure, we can actually go beyond the boiling point of water, and we reach this 121 degrees Celsius temperature. And this 121 degrees Celsius is a very, very high temperature, and these growth factors are heat sensitive and they will get degraded at this high temperature. That's why we do not autoclave the mammalian cell culture medium. We always filter this medium. We uh, we have these 0.22 micron filters, and if you go to the lecture video, we have a demonstration where like how these mammalian cell culture mediums are media is prepared and it is filtered through these filters. Okay, so that's why we don't use the autoclaving process. For bacterial medium, for bacterial cell culture medium, we generally use autoclaving process because in that medium, the components that are added to the medium are not heat sensitive generally. For some bacterial cell culture medium also, we prefer filtration over autoclaving. Like some of the minimal salt medium, uh, we have different salts. So when we heat them and we cool them, sometimes some of the salts precipitate. So it's better to filter sterilize it than autoclaving it. Okay. So basically, if your uh, medium is stable in the basically if your medium is stable at a high temperature, then you can autoclave it. Otherwise, it's always better to filter sterilize it. Okay. Option C, that is filtration is cheaper, is absolutely wrong. It is actually more expensive than autoclaving because you have to buy those 0.22 micron filters and they are not very cheap. Okay. So autoclaving is obviously a cheaper option, but if things are heat sensitive, you cannot autoclave it. Okay, you have to go for the filtration method. I hope that's clear to everyone. If you have any doubt, you can write in the chat box or you can unmute your mic and ask. Okay. Moving on to the next question now, question number 7. Why is pH meter not used for checking the pH of DMEM media? Option A, media components might block the pH probe bulb. Option B, media might get contaminated. Option C, pH meter does not work inside DMEM media. And option D, pH strips are more precise than pH meter. Why is pH meter not used for checking the pH of DMEM media? Media components might block the pH probe bulb. Media might get contaminated. pH meter does not work inside the DMEM media, and pH strips are more precise than pH meter. What is the correct answer for this? What is the correct answer? Come on, who wants to answer? Why is pH meter not used for checking the pH of DMEM media? It's a mammalian cell culture media. I just explained what is a DMEM media, why are we adding different components in the DMEM media. So how can we check the pH of this DMEM media? Okay, we have one answer from Rosie. pH strips are more precise than pH meter. Okay. Anyone else wants to answer? Okay. We have another answer from Nilina. That is also option D. So actually the major reason why we don't use pH meter is because media components can block the pH probe bulb. So pH meter basically like in a pH meter we have a bulb where when the ions are interacting, it can basically measure the hydrogen ion concentration and then Based on the current, we get the pH reading, right? So, since this DMEM media has several components, we have the FBS, that is the fetal bovine serum. So it has multiple growth factors. So many different components are added in the medium. So these components actually hinder with this pH measurement. Okay, and we don't get very accurate measurement with the pH meter. That's why we go for pH strips. So in general, pH strips and pH meter both are very precise, but in this case, since the media components block the pH probe bulb, the pH strips are actually giving a better reading than pH meter. pH meter cannot uh, give an accurate reading. So pH strips are not generally more precise than pH meter. pH meter is obviously more precise because you have in a pH meter you can check the pH up to 2 decimal points, or if your instrument is more sensitive you can see up to 4 decimal points also. But and in a pH strip it is general, it just gives you a general idea because it is color based and it is very subjective, because like if I add a drop of media to the pH strip and then check the color, it is very subjective. Like the color I am perceiving, maybe you perceive the color differently and you can report the pH to be slightly different than my observation. Okay, so in that sense pH meter is more precise. But for measuring the pH of DMEM media, since the media has multiple components in it, it is actually those components can actually block the pH probe bulb, and that's why we don't add the pH probe inside the medium. Okay, this is clear to everyone. Do you guys understand why option D is incorrect? Yeah, okay, great. So I'll move to the next question now. Question number 8. Why is serum heat inactivated before putting in the cell culture medium? Why is the serum heat inactivated before putting in cell culture medium? Option A, to sterilize the serum. Option B, to dissolve it in media. Option C, to inactivate the complements in the serum. And option D, to activate the complements in the serum. Why do we heat inactivate the serum before putting it in cell culture medium? Anybody knows the answer? Do we heat inactivate to sterilize the medium, to dissolve it in the media, to inactivate the complements in the serum, or to activate the complements in the serum? Yeah, we have one answer, option C, to inactivate the complements in the serum. Okay. Anyone else wants to answer? Okay, Rosie says option D, to activate the complements in the serum. Okay. So the correct answer is to inactivate the complements in the serum. Yeah, so two people answered option C. Option C is the correct answer actually. So basically, I told you what serum is. Serum is the liquid part of the blood. So if you have blood in a vial and you let it sit for some time, okay, what will happen? Sorry, my video is off. Yeah. So if you have blood in a vial and then if you let it sit for sometimes, whatever clotting factors are there, so they will clot the RBCs, they will settle down, and the liquid part, the yellow whitish liquid part that you will see on the top will be serum. Okay. So serum is basically the liquid part of the blood. Now that serum has multiple growth factors. That's why we use it in the cell culture medium. But along with these growth factors, they will have multiple complements. Now these complements, when we add in the growth medium, they will actually attack the cells, and the growth of the cells will be inhibited because of those complement. Because now that serum belongs to a particular organism. So generally we use fetal bovine serum. So it is a, fetal bovine whose blood serum is taken. Okay, so it belongs to a particular organism. Now whatever cell line I am growing, that can belong to us, that can, and that will belong to some other organism. So whatever complements that are present in the serum, they can react with the cell line that you are growing, and they can actually inhibit the cell growth. To prevent that in cell growth inhibition, because we are adding the serum as a growth factor source. So if we, sorry, so if we add the serum and it is not helping in the growth, instead it is actually inhibiting the growth, our purpose is not solved. So for that we have to inactivate those complements. So how do we heat inactivate the serum? We basically, some mostly commercially available fetal bovine serum is already heat inactivated, so you can directly use it in your medium. Other than that, you can heat inactivate the serum on your own by simply heating it, and let the complements precipitate down, and then you can just filter the serum and use it in your medium. Okay, so that is a simple heat inactivation process, but mostly commercially available serum are already heat inactivated. You can buy that and directly use it in cell culture medium. But why heat inactivation is important? It is important to inactivate those complements in the serum that are going to inhibit the cell growth, which we do not want. We are adding the serum to enhance the cell growth, not inhibit the cell growth. So if something is inhibiting the cell growth, we will have to uh, make sure that that is not happening. Okay, I hope that's clear. Rosie, is that clear? You answered option D, so I hope this is clear why option C is the correct answer. Great. So we'll move to the next question now, question number 9. If you guys have any doubt, you can write it in the chat box and I'll try to solve it. Okay, so question 9. Dash media is used for the production of baculovirus in Lepidopteran cell lines. Option A, modified IPL 41 insect medium. Option B, TC100 medium. Option C, DMEM media. And option D, Hinks TNM FH medium. Dash media is used for the production of baculovirus in Lepidopteran cell line. Anybody knows the answer for this? Or anybody wants to answer this? Anyone? Okay, I'll tell the answer for this. This is a memory based question, so it's okay if you don't know. So the correct answer for this question is option B, TC100 medium. So this production of baculovirus and lepidopteran cell lines, so it's a insect cell line culture. So right now we were studying about DMEM medium that was used in mammalian cell culture. So cell lines that are obtained from mammals. Okay. Similarly, there are cell lines that are obtained from different insects. So those cell lines, for those cell lines, we have a different growth medium, because insects have a different uh, growth requirement. Okay, so bacteria have different growth requirements, so we have a separate medium for that. Then mammals have different growth requirements, so we have a different growth medium for mammalian cells. Then insects have a different growth requirement, so we have different medium or different media for different insect cell lines. Okay, so this is a list of uh, different medium that are present, that are available for different kinds of insect cell lines. So we have these SF9, SF21 cell lines for which we use this Grace's insect medium supplemented. And then we have this Trichoplusia Ni cells, which have this Hinks TNMFH insect medium, like that is used for these cell lines. Then we can also use IPL41 medium for SF9 and SF21 cell line. For production of baculovirus and lepidopteran cell lines, we use TC100 medium. For some mosquito cell culture, we have a different medium. Then for Drosophila, we have different medium. So for different insects, we have different medium. Okay. Moving on to the next question, question number 10. Which of the following is a characteristic feature of eukaryotic cells but not prokaryotic cells? Option A, presence of ribosome. Option B, nucleus. Option C, cell wall. Option D, circular DNA. Which of the following is a characteristic feature of a eukaryotic cells but not the prokaryotic cells? Which is a feature for eukaryotic cells but not prokaryotic cells? Anyone wants to answer? Come on, this is a easy question. Do you know the difference between eukaryotic and prokaryotic cells? If you know the difference, you should be able to answer this. Which of the following is the characteristic feature of the eukaryotic cell? Like something that is present in eukaryotic cell but not in the prokaryotic cell. Presence of ribosome, nucleus, cell wall, or circular DNA. Okay, Rosie has answered option A, presence of ribosome. Anyone else? So are ribosomes present in both eukaryotic and prokaryotic cells, or is nucleus present in both eukaryotic and prokaryotic cells? Okay, we have two more answers. Option B, that is nucleus. Yes, that is the correct answer actually. So nucleus is present only in eukaryotic cells and not prokaryotic cells. So previously also I have explained the difference between a prokaryotic cell and a eukaryotic cell. So a prokaryotic cell, you can consider it as a one room. Okay, so in a single compartment, there are no compartmentalization inside the cell. So this is a prokaryotic cell having the cytoplasm. So the cytoplasm is there all over the cell, and then things are just floating. All the proteins and all the genetic material, everything is floating in the cytoplasm. Okay, so there is no separate membrane bound organelles. Whereas eukaryotic cell, you can consider it as a apartment where different rooms are present. So different organelles, membrane bound organelles are present in the eukaryotic cell. So inside the cell, you will have different membrane bound organelles. So you will have a nucleus, well-defined nucleus with its own membrane, and inside the nucleus you will have the DNA. So you will have the DNA inside the nucleus. So this will be a eukaryotic cell. And in prokaryotic cell, you will have the DNA floating in the cytoplasm. So this is cytoplasm. Here also we have cytoplasm, but in the cytoplasm different organelles are also present which have their own membrane. Okay, so that means this is the prokaryotic cell, prokaryotic cell, and this is a eukaryotic cell. Okay, so ribosomes are present in both eukaryotic and prokaryotic cell. Cell wall is present. Cell wall is also present in both eukaryotic and prokaryotic cells. So not all eukaryotic cells have a cell wall, but uh, plant cells have a cell wall, and most of the prokaryotic cells have a cell wall. Bacteria has a cell wall made of peptidoglycan. Okay, then some prokaryotes have another layer, another protective layer called capsule. Nucleoid that you see, this is just a region where the DNA is present. Okay, it's just the region where the DNA is present. It doesn't have a defined medium, defined membrane around it. So there is no membrane present around the DNA. So this is just the region where the DNA is localized. Whereas in a eukaryotic cell, you can see the nucleus has its own membrane. It has its own nucleolus. Like you can see here, you have the nuclear membrane, then you have the cytoplasmic membrane, and you have different organelles. The chloroplast is there, Golgi body is there, mitochondria is there, endoplasmic reticulum is there. Ribosomes are present in both eukaryotes and prokaryotes. In prokaryotes, ribosomes are freely floating in the cytoplasm, whereas in eukaryotes, the ribosome is generally attached to the endoplasmic reticulum, as you can see here. Okay, these are the mitochondria, and then you have the cytoplasm in which all inside which all these uh, organelles are present, all these membrane bound organelles are present. So presence of ribosome, it is there in both eukaryotic and prokaryotic cells. Cell wall is also there in both eukaryotic and prokaryotic cell. Circular DNA is a characteristic feature of prokaryotes. It is a characteristic feature of prokaryotes. Okay, so in eukaryotes we have linear DNA, in prokaryotes we have circular double stranded DNA. Okay, this is clear to everyone. Moving on to the next question now, question number 11. Dash allows for mobility of the prokaryotic cell. Option A, flagella. Option B, phospholipid. Option C, cytoplasm. And option D, lipopolysaccharide. What is present in the prokaryotic cell that allows its mobility? Okay, we have one answer from Vaishnavi. Option A, flagella. Okay, Rosie has also answered option A, flagella. So that is the correct answer. Option A, flagella allows for the mobility of the prokaryotic cell. So here in this figure, you can see there's a single flagellum present. Flagellum is the singular, and once you have more than one flagellum, you can call it flagella, that is the plural. So flagella basically allows the cells to move. Okay, so for humans we have our hands to swim, right? So these prokaryotes, they are very tiny cells, right? And it's just one compartment. It does not have any arms or legs to organs to facilitate its movement. We have the legs to walk, we have our hands, we can use our hands and legs to swim in water, we can walk on land. But for bacteria it is not possible. So for bacteria, the movement is facilitated by this special structure called flagellum. So flagella basically allows the bacteria to swim in the medium, swim around in the medium, or move around in the medium. So it can use the flagella as a propeller to, or a glider to glide in the medium. Okay. So like we use our hands to swim in water, similarly bacteria prokaryotes are using flagella to swim in the environment. Okay. So flagella allows for mobility of the prokaryotic cell. Mobility means movement. I hope that's clear. Moving on to the next question, question number 12. The region between inner layer and outer layer of gram-negative prokaryotic cell membrane is called as option A cytoplasmic space, option B phospholipid, option C periplasmic space, and option D lipopolysaccharide. So what is the space between the inner and outer layer of the gram-negative prokaryotic cell membrane called? Anyone wants to answer? What do we call the space between the outer and the inner cell membrane of a prokaryotic cell? Okay, Vaishnavi answered option C. Anyone else wants to answer? You've answered periplasmic space, option C. Okay. Anyone else wants to answer? Okay, we have one more answer, option C. So option C is the correct answer, periplasmic space. So in a gram-negative prokaryotic cell, we have two layers of membrane. So we have the cell. So this is the outer membrane. And then we have a inner membrane. Okay. So the part inside the inner membrane, this is called cytoplasm. Okay. And then the part between the inner and outer membrane, I'll show it with a different color. So the part shaded with green here, so this is the periplasmic space. Okay. So if I say that a particular protein is present in the periplasm, that means it is present in this region. Okay. And if I say this protein is cytoplasmic, that means it is present in the center region inside the inner membrane. Okay. So this is the outer membrane. And then this is the inner membrane. Okay, so this is the inner membrane, this is the outer membrane. The space between the inner and outer membrane is the periplasmic space, and the space inside the inner membrane is the cytoplasmic space. Okay, So the difference between cytoplasmic space and periplasmic space is clear to everyone. Is it clear to everyone? The difference between the cytoplasmic and periplasmic space. Okay, so the space between the inner and outer membrane is periplasmic space. The space inside the inner membrane is the cytoplasmic space. Okay. Phospholipids are just the units that are composed in the membrane bilayer, and this is present mainly in the eukaryotic cells. So option C, periplasmic space, is the correct answer here. Okay. Moving on to the next question now. Question number 13. The most common pH range for cell culture media is? What is the most common pH range for cell culture media? Option A, 5.5 to 6.5. Option B, 7 to 7.4. Option C, 8 to 8.5. And option D, 9.9 to 9.5. So what is the most common pH range for cell culture media? 5.5 to 6.5, 7.0 to 7.4, 8.0 to 8.5, 9.0 to 9.5. Okay, Rosie has answered option B, 7.0 to 7.4. Anyone else wants to answer? Okay, we have one more answer from Runmai. Option B, 7.0 to 7.4. So yes, that is the correct answer. So for cell culture medium, we generally maintain the pH at 7.0 to 7.4 unless there is some special requirement from a specific cell line. So all the mammalian cell, as you know, like our inside our body also, this is the pH that is maintained by our body. So when we are culturing that cell outside, we need to maintain that particular pH. And I have already explained how the pH is maintained by sodium bicarbonate and CO2 present in the like medium. CO2 is provided through CO2 incubators, and sodium bicarbonate is already part of the DMEM media. So they both combine to make a buffering system where they are able to maintain the pH at 7.0 to 7.4, okay, in this range. This is clear? Is this clear to everyone? So option B is the correct answer. Moving on to the next question. What is the role of phenol red present in the mammalian cell culture medium? What is the role of phenol red present in the mammalian cell culture medium? Option A, to check the temperature. Option B, to monitor the pH of the medium. Option C, to observe growth of the microbial contamination. And option D, to check CO2 levels. Okay, we have one answer. Option C, to observe growth of the microbial contamination. Okay, anyone else wants to answer? Why do we add phenol red to the mammalian cell culture medium? Why is phenol red added to the mammalian cell culture medium? Anybody? Yeah. Okay. We seem to have different answers from different people. So Rosie has answered option C, to observe the growth of microbial contamination. Mrinmay has answered the same thing, and Vaishnavi has answered to monitor the pH of the medium. So option B is the correct answer. So Vaishnavi has the correct answer, to monitor the pH of the medium. So phenol red is a pH indicator. Okay. So phenol red is a pH indicator. And what do we mean by pH indicator? So these solutions have a property of changing its color at different pH. So basically phenol red is red in color, red or pinkish in color in the pH range of 7 7 and above. Okay. Below 7, the color will be yellow. Okay. So here the color will be red or pink when the pH is 7 or above 7. And when the pH is lowered, when the pH becomes acidic, when the pH decreases from 7, the medium starts turning yellowish. Okay. So I've told you previously also, when we are growing a mammalian cell line in a medium, in a cell culture medium, some cells are growing, some cells are dividing. Along with that, we will have some cells that are dying. Okay. Once the cell dies, it will get lysed, it will burst open, and the components that are present inside that dead cell will come out. Now those components can actually make the medium acidic. And if for some reason our cell culture medium is not able to buffer the system properly, like the sodium bicarbonate and the carbonic acid buffer system is not working properly, or if the added acid is very high in concentration, like if some components are released by the cells that are too acidic, and our buffer system is not able to maintain the pH at 7, the medium will turn from red or pink to yellow immediately. Okay. So when you are growing, so the medium is already having phenol red, so medium looks reddish in color. Okay. So you can Google DMEM uh, cell culture medium, and you will see the color of the medium is reddish or pinkish red, dark pink or red in color. Okay. And when you inoculate the cells and you let the cells grow in that medium, uh, and let's say there's some contamination, or there's some factor that is released by the cell that is acidic, you will see the medium has turned yellowish from red pink color. Okay. That red pink color that is there, that is present in the medium, you will see the color has changed from red to yellow. Okay. And that change in color will immediately tell us that okay, the pH of the medium is not maintained. That means we have to add certain components to the medium to maintain the pH, or we have to maybe increase the CO2 percentage that we are providing the cells in order to maintain the pH. Maybe the cells are not able to maintain the pH at that particular CO2 percent. So if you are giving 5% of CO2, maybe the medium is not able to buffer the system, and you might have to increase the percentage of CO2 in order to uh, maintain the pH at pH 7 to 7.4, as we looked in the previous question. Okay. So if your medium is turning yellow, that means something is wrong, something is there that is changing the pH of the medium, and you have to immediately work on, like, figure out what is changing the medium, and what is changing the pH of the medium, and you have to change that particular factor so that your pH remains 7 or 7.4. Your medium should look red or pinkish in color. This is clear to everyone? So both of you have answered option C, to observe growth of microbial contamination. It is, you can say, indirectly correct answer if there is some other contamination that is acidic in nature. So generally fungal contaminations, fungus secretes a lot of acidic components. So uh, fungal contaminations also tend to give a yellowish color to the media because of the acids released by the contaminants. But mainly we add the phenol red to as a pH indicator. So by looking at the pH, then whatever is your conclusion, if you're concluding that okay, we have a contamination, that's why my pH changed, or my cells are secreting something, that is why my pH is changing, that depends on your experiment and your observation. But phenol red is added to observe the pH, to monitor the pH, whether the pH is remaining at 7 to 7.4 or the pH is getting decreased, or uh, by release of some acidic components. Okay, this is clear. Both of you have answered option C. I hope this makes sense to you now. Okay, great. Then I'll move to the next question. Question number 15. In gram-negative bacteria, porins are present in dash. In a gram-negative bacteria, porins are present in dash. Option A, outer membrane. Option B, periplasmic space. Option C, cytoplasm. And option D, phospholipid layer. In a gram-negative bacteria, where are these porins present? Do you know what porins are? Anyone knows what porins are? Anybody? Anybody knows what porins are? What are porins? So porins are basically channel proteins. Okay, these are proteins that have pores in them that can facilitate. Yes, Rosie has answered. So they are channel proteins. So they have pore. They have a pore that can facilitate the entry and exit of certain uh, ions or certain components, certain factors that are needed by the cell. So if the cell needs something and is present outside, it can form a channel, like these porins form a channel that can facilitate the movement of molecules through the protein. Okay, so where are these presents? Mrinmay has answered option A, outer membrane. Yes, that is the correct answer. So it is present in the outer membrane. So I told you that these are there for transportation of molecules, right? So they are present in the outer membrane and they are like a pore. So basically we have the membrane like this, and then we have these porins that are basically hollow. So this is my membrane. So these porins have the hollow center that can facilitate molecules to pass from the outside. This is the outside and this is the inside of the cell. Okay, and these are the porin proteins. Okay, these are protein channels. Okay, these are protein channels that facilitate the movement of a molecule from outside to inside. Okay, so these are embedded in the outer membrane to facilitate the movement of any molecule from outside to inside the cell. This is clear. Moving on to the next question now. Bacterial cell wall is composed of dash cellulose, option A, option B, peptidoglycan, option C, chitin, and option D, exopolysaccharides. Bacterial cell wall is composed of what? Option A, cellulose, option B, peptidoglycan, option C, chitin, and option D, exopolysaccharide. Okay, we have answers from two people saying option B, that is peptidoglycan. Yes, that is the correct answer. So bacterial cell wall is composed of peptidoglycan. Peptidoglycans are basically polymers of glycan. So we have two types of glycans, mainly N-acetyl glucosamine and N-acetyl muraminic acid. So NAM and NAG units. We have alternating units of these glycans, and these glycans are attached with peptides. That's why the name peptidoglycans. So we have peptides, short peptides, like 6 amino acid peptides or 4 amino acid peptides. I don't remember exactly. So these short peptides are added to the glycan molecules. Glycans are basically carbohydrates. Okay. So these glycan molecules are attached to these short peptides, and these glycan molecules form a polymer kind of a structure that is present in the bacterial cell wall. So this peptidoglycan is the building block of the bacterial cell wall. Okay. Cellulose is generally present in the plant cell wall or some of the fungal cell walls. Chitin is also present in the fungal cell wall. Okay. An exopolysaccharide is present outside the cells. It's not part of the cell wall. It generally forms a layer outside the cell. Okay. So peptidoglycan is the bacterial cell wall component, and it is called peptidoglycan because it is glycan polymer to which short peptides are added. So peptidoglycan. Okay. So all of you have answered option B. So I hope this is clear. I'll move to the next question now. Which of the following statements are true for handling of cell and culture media? First statement is cells and culture media should be free from contaminating microorganisms. Second statement is all cell culture materials should be maintained under sterile conditions. And third statement is all surfaces within the cell culture area should be non-porous to prevent adsorption of media and other materials that may provide a good breeding ground for microorganisms, resulting in infection of the culture. Okay, these are the three statements. Which of the following statements are true for handling of the cells and culture media? Option A, first statement only. Option B, second and third only. Option C, first and third only. Option D, all of the above. So all of the above statements are true, or first and third statement is true, second and third statements are true, or only first statement is true. So first statement is cell and culture media should be free from contaminating microorganisms. Second statement is all cell culture material should be maintained under sterile conditions. Statement 3 is all surfaces within the culture area should be non-porous to prevent adsorption of the media that may provide a good breeding ground for the microorganisms, resulting in infection of the culture. So what would be the answer? Which of the following statement is true? You have to identify the true statements. So are all three statements true? Is the first statement only true? Is the second and third statement only true? Or first and third statements are true? Which of the following option is correct? Come on, answer. Which statement is correct here? No one knows the answer for this. It's not that difficult. Think logically. Do you think the cell culture media should be free from contaminating microorganisms? Do you think the statement is true or false? Or do you think things should be maintained under sterile condition? Yeah, we have one answer from Runmai. Option D, all of the above. Okay, very good. Anyone else wants to answer? Okay, Rosie answered option A, that is first statement only. Okay. Vaishnavik, do you also want to answer? Do you agree with Mrunmay or do you agree with Rosie? Yeah. So option D, you have also answered option D. So we have the majority for option D. I can say that it is the correct answer. So all of the above is actually required for cell culture handling. So your media should be free of contaminating microorganisms. So I've talked about sterilization processes. So I've talked about filtration and autoclaving, two sterilization methods. So sterilization is important to prevent any contaminating microorganism. So let's say I want to grow a particular bacteria. Let's say I want to grow E. coli. Okay. Now, if my media already has other organisms, spores of other organisms, like potential my media has the potential to grow other organisms. Okay. So now when I'm culturing E. coli, there will be other things that are floating in the media along with E. coli, right? And I don't want that. I want a pure culture of E. coli. So for that, what will I do? I will first sterilize my media by autoclaving it or by filter sterilizing it, and then I'll use. So my cells and the cell culture media, they should be free of any contaminating microorganisms. They should be pure, pure in the sense like the cell line should be pure. It should contain only one type of cell, and the media should be free of any microorganism. It should not have anything until I'm adding the inoculum, which is also containing only one type of cell. So my media is also containing only one type of cell. Okay. Then all cell culture material should be maintained under sterile condition. That is also necessary. Now I've autoclave, like I've sterilized my media. I have the cells that are consist that consists of only one type of cells, but the micro tips that we are using, the pipettes, the uh, other things that we are using, if we are using a syringe to add something to the media, so whatever you are using in the uh, along while doing the cell culturing, they also have to be sterile. They also have to be sterilized by some process. Otherwise, they can also cause contamination of your medium. Okay. Then the third statement, that all surfaces within the culture area should be non-porous to prevent adsorption of the media and other materials. This is actually very important, because if you're let's say the hood that you are using, the cell culture hood where you are working, that has some surface that is absorbing the media. So let's say by mistake some of the media components got spilled. Okay, I spill the media, and the media is now absorbed in the on this hood surface. So it can actually provide a breeding ground for other microorganisms. Now microorganisms can obviously eat these media components because they have all the growth uh, factors that are required. That is why we are using it for cell culture medium, right? We are using these as a growth medium because they can allow the growth of microorganism. But we want to grow a particular microorganism, or we want to grow a particular cell, but other microorganisms that are present in the air, they can also grow in that. So to prevent the growth of these microorganisms on the in the culture area, we should have non-porous materials. We should not have porous materials that can absorb the media components or something that can provide a growth surface for these microorganisms. Okay, so otherwise they can also result in contamination or infection. So this is clear. So all the statements are actually true for handling cells and culture media. I hope that's clear. Move to the next question now. What is the purpose of the cell fractionation? Option A, it allows cellular components such as organelles to be studied in isolation. Option B, it allows cellular components to be viewed in a light microscope, viewed with a light microscope. Option C, it allows for the study of interactions between all the organelles. And option D, mitochondria and endoplasmic reticulum cannot be studied together. What is the purpose of cell fractionation? Why do we do cell fractionation? It allows cellular components such as organelles to be studied in isolation. It allows cellular components to be viewed with a light microscope. It allows for the study of interaction between all the organelles, or we do cell fractionation because we cannot study mitochondria or ER together. What is the reason for doing cell fractionation? What is the purpose of doing cell fractionation? Okay, Vaishnavik has answered option D. Okay, anyone else? Rosie has answered option A. It allows cellular components such as organelles to be studied in isolation. Simranmai, with whom do you agree? Is it A or is it D? Okay, you are also saying A. Again, we have the majority for option A, so I'll also go with the option A. It is the correct answer actually. We do cell fractionation in order to separate different organelles and different cell components to be studied in isolation. That means we can separate nucleus, then we can separate the mitochondria, then we can separate the endoplasmic reticulum, we can separate the ribosomes. That's how we can, like if I want to study mitochondria, then to study the mitochondria in isolation, to isolate the mitochondria, we can do cell fractionation where we can separate different cellular components and we can take out each component separately, isolate each component separately and then study them. Okay. So mitochondria and ER can be studied together. There's no restriction in that. But if I want to study ER separately and mitochondria separately, I can do cell fractionation to study them in isolation. Okay, so to separate any cellular component from the rest of the cell, we can do cell fractionation where we can uh, separate different components and step by step to isolate each and every component and then study each component like that. This is clear. So option A, it allows cellular components such as organelles to be studied in isolation is the correct answer. Vaishnavik, is it clear? You have answered option D. I hope the explanation is clear to you. Great. So moving on to the next question now, question number 19. The first step in cell fractionation is typically option A homogenization, option B centrifugation, option C filtration, and option D dialysis. What is the first step for cell fractionation? Homogenization, centrifugation, filtration, or dialysis? What will you do first? If I give you a tissue sample, or if I give you a any kind of sample, if I give you a liver sample, what will you do? What will your first step while doing the cell fractionation? Okay, Mrinmay has answered option A, that is homogenization. Okay, Rosie has also answered option A. Okay, so all of you seem to agree with option A, that is homogenization. So that is the first step, that is the correct answer. That is the first step we take while we are doing cell fractionation. So if I give you any tissue sample, or if I give you any let's say a liver sample or a muscle sample, and I ask you to fractionate the cell, like take out the nucleus separately, take out the mitochondria separately, take out the different organelles separately. So what will you do? First, you have to homogenize the cell. Homogenization means you have to uh, kind of break down the cell and solubilize it in a particular medium, okay, particular buffer, where you can actually then you can actually do the centrifugation and further process to separate the different fractions, right? So for doing centrifugation, you can't directly put the liver in the centrifuge and centrifuge it, right? You have to dissolve it and homogenize it. You have to make a homogeneous mixture. How do I explain? You have to make a continuous mixture for a solution, a clear solution of the whole sample that I have given. So for that, the first step is homogenization. Once you have a solution of that cell where the cells have broken down, so now you can take that solution and you can do the centrifugation for collecting different fractions. So that whatever approach you are taking for cell fractionation, that technique can be applied after homogenization. Okay, so option A is the correct answer. Moving on to the next question now. Which step of cell fractionation procedure separates the cell into its fraction? Which step of cell fractionation procedure separates the cell into its fraction? Option A, centrifugation, option B, collection, option C, lysing, and option D, purification. So which of the following step of the cell fractionation procedure separates the cells into its fraction? Okay, so we have one answer, option A, that is centrifugation. Okay, others can also answer. Which step of cell fractionation procedure separates the cells into fractions? Yeah, so all of you seem to agree with option A, that is centrifugation. So as I have explained, first you will do the homogenization, and then the next step would be centrifugation, where you can separate different cell components into different fractions. You can separate the nucleus, you can separate the membrane, you can separate the mitochondria and the ribosomes. So like that, you can have different fractions and then study each and every fraction separately. Okay. Sorry. Yes, so option A, centrifugation, is the correct answer. Moving on to the next question. The first centrifugation step in differential centrifugation typically separates Option A, nuclei and cell debris. Option B, mitochondria and lysosomes. Option C, endoplasmic reticulum and Golgi apparatus. And option D, ribosomes and small molecules. So the first centrifugation step in differential centrifugation typically separates what? Option A, nuclei and cell debris. Option B, mitochondria and lysosomes. Option C, endoplasmic reticulum and Golgi apparatus. And option D, ribosomes and small molecule. What is the correct answer here? What do you think is the correct answer here? So Rosie has answered option A, nuclei and cell debris. Yes, both of you have answered nuclei and cell debris. That is the correct answer, option A. So this is what I was talking about. So once you have the, let's say, a tissue sample, you will put it in a buffer, like slice it and chop it off and then put it in a buffer and then do homogenization, where everything will be, all the cells will be broken apart and they will be in the solution. It will be in a homogeneous solution, and then that solution will have different components. They will have the nucleus, they will have the mitochondria, lysosomes, plasma membrane, and then they will have the ribosomes, viruses, macromolecules, everything. So now to separate each component separately, basically first you want to remove the nucleus. So you spin it at the very low speed, 600 G, for 10 minutes. So only nucleus will settle down and rest of the things will be in the solution, because nucleus is the heaviest uh, component in that mixture. So nucleus has the genetic material, so it becomes the heaviest component of that mixture. So nucleus and the cytoskeleton, they will settle down and rest of the things will float. Then in the next step, you can again take that particular supernatant. So here, after spinning, we will take the supernatant in a separate tube and then spin that in at 15,000 G for 15 minutes. When you do that, you will see the pellet that you will get. Mitochondria, lysosomes and peroxisomes will settle down and rest of the things will again float in the supernatant. Again take that supernatant and spin it at further high, like higher speed, that is 1 lakh G for 60 minutes, that is 1 hour. So you spin the tube for 1 hour at 1 lakh G, and you will see the plasma membrane, the ER fragments and the small vesicles, they will settle down and rest of the components will be floating in the supernatant. Then you take that supernatant and spin it at even higher speed, that is 3 lakh G, and for spin it for 2 hours. You will see all the lightest material that is present, that ribosomes and macromolecules that are present, that will settle down, and then the supernatant that you will get will be the cytosol. Okay, cytosol means cytoplasm. Okay, so this is the process. So the first thing that will fraction out will be, nucleus and cytoskeleton. Next, the so the heaviest component is nucleus and cytoskeleton. Then we will have mitochondria, lysosomes and peroxisomes that will settle down, and then we will have plasma membrane, ER fragments and small vesicles that will settle down, and then the lightest material, that are ribosomes and different viruses and macromolecules, they will settle down. Okay, so this is how we do fraction differential centrifugation. So we are taking the sample, centrifuging it, again taking the supernatant, centrifuging it again at a higher speed, and then again taking the supernatant, and then we are centrifuging it again at a further higher speed. So we are centrifuging at every step at a by increasing the centrifugation speed. Okay, so that is a process of differential centrifugation. So we are doing differential centrifugation to separate different components present in the tissue. This is clear? Okay, moving on to the next question. Question number 22. What is the principle of centrifugation? So we have talked about differential centrifugation where things are settling. So what do you think the principle behind centrifugation is? Option A, sedimentation principle. Option B, filtration principle. Option B, evaporation principle. Or option D, size reduction principle. What is the principle of centrifugation? Tell me, what do you think will be the answer? Yeah, option A, sedimentation principle. Yes, both of you are correct. Option A, sedimentation principle is the correct answer. So basically, when you are doing centrifugation, so in physics you must have studied about centrifugal force and centripetal force. Okay, so if you are spinning something at a fixed axis, around a fixed axis, if you are spinning something, so you have the sample here and you are spinning this sample around a fixed axis in a circular motion. So there will be two forces, a centripetal force towards the center and a centrifugal force away from the center. So the force that is away from the center is applied in centrifugation. So you have tubes. So let's say you have a tube here which has a solution, and that solution has different components. So components that will be heavier. So let's say the components like uh, so components that are heavy in nature, that has a higher sedimentation rate, okay, that have a higher mass or higher like that are bigger in size, that will have a higher centrifugal force. Okay, now that if we have two components, component 1 and component 2, let's say the component 1 is heavier than the component 2, what will happen? The centrifugal force will force the component 1 to settle down first. So when we will centrifuge, we will get the pellet of component 1 and we will get the supernatant which will have the component 2. Okay, so basically based on the speed of centrifugation you can control which component you want to settle down. That is what we were doing in the differential centrifugation, where at low speed we were settling the heavier components down, at a higher speed we were settling the lighter components down. Okay, at some or the other speed you will have everything settled down from the solution. It's just that it depends on your equipment's efficiency, how like what is the highest speed limit for your equipment. Okay, okay, okay. Next question is, principle behind density gradient centrifugation is: option A, particles move through a gradient until they reach a density equal to their own. Option B, particles are separated based on their size and shape. Option C, particles are separated based on their charge. And option D, particles are separated based on their solubility. Principle behind density gradient centrifugation is: what is the principle behind density gradient centrifugation? Option A, particle move through a gradient until they reach a density equal to their own. Option B, particles are separated based on their size and shape. Option C, particles are separated based on their charge. And option D, particles are separated based on their solubility. Okay, Rosie has answered option B, particles are separated based on their size and shape. Okay, for density gradient centrifugation, what is the principle? So for any centrifugation, the particles are separated based on their size and shape. But for density gradient centrifugation, there's a particular factor that is associated, that is like a particular property that is exploited. to separate these particles? Yes, option A. Mrunmayee has answered option A. That is the correct answer. So, what we have in density gradient centrifugation is we have a gradient of different densities. So, generally sucrose solution is used. So, different concentration of sucrose solution will have different densities, right? So, here we have a figure. So, so this is a solution of sucrose with different density. So this part will have a different density. It's a higher concentration of sucrose. Then this part will have a different density. This part will have, and it's a continuous gradient. It's not a step gradient. So the gradient is increasing. So this is low to high. So the gradient is increasing as we go down. The density gradient is increasing. So this is the most dense and this is the least dense sucrose solution. Now we load the sample, the mixture that we want to separate. So when we start centrifugation, so in this technique, in density gradient centrifugation, the centrifugation speed is constant. Okay, the speed of centrifugation is constant. Okay, the speed of centrifuge is constant. What is different is density. The separation is based on the density here. In differential centrifugation, the speed was the factor that was changing. We were increasing the speed. So based on the size of the molecule, higher speed means the like lower speed, only heavier particles can settle. When we increase the speed, higher particles were settling down based on their sizes. But in density gradient centrifugation, we have a gradient of density from low to high. Now, when we are doing the centrifugation at a constant speed, these molecules will be separated based on their density. So, let's say this yellow molecules have certain density, then gray molecules have a certain density, and blue molecules have a certain density. Okay. Now, the blue molecules being the heaviest, they will, like heaviest in the sense of density, they are the most dense, so they will move till they match the density of the sucrose solution. Wherever the density of these blue molecules will be equal to the density of the sucrose, they will form a layer there. Okay, then the gray molecules will form another layer based on their density, and then the lightest molecule, that is the yellow molecules, they had the least dense, they were the least dense molecules. They settle at the top part. Now, once you have these layers separated, you can either pipette these layers out, or you can have a opening at the bottom and you can collect these layers. So you can collect the fraction till here, you will have only the blue components. Then you can collect the fraction till here, you will have the gray component. Then you can collect the fraction till here and you will have the yellow component separated. So that's how you can separate the components based on their densities. Okay, so when the particles move through the gradient, they will move till they reach their own density. They will reach the density equal to their own and they will stop at that point. Okay, so this is clear to both of you. So this was the last question of this session. I hope principle behind differential centrifugation, density gradient centrifugation is clear. Principle behind sterilization, why we are using different components in a cell culture medium, that is clear. And what else did we learn? The difference between synthetic and like defined and the complex medium. So these are the things that are covered in this session. So if you have any doubt, you can ask me. I can try to clear it off. Any doubt? Okay, great. You can, I will post the recording of this video so you can go through the recording also. And if you have any doubt, you can let me know in the next class also. So thank you so much for joining. I'll see you in the next class now. I'll end the call now. Thank you.